Okay, so last time they call the Starling principle, and it's illustrated by these Starling curves. So along the x-axis, you can see right atrial pressure. Now, this is a diagram that was developed for humans or other higher vertebrates. So in fish, there is no such thing as a right or left atrium, but the concept is still the same. So the atrium pressure or the end diastolic volume. So the amount of volume that's remaining in the heart, specifically in the atrium, after contraction of the ventricle. So as that increases, in other words, as the atrium has additional blood ready to be delivered to the ventricle for its next contraction, as that increases, the stroke volume or cardiac output, the amount of blood that's delivered by the ventricle with its next contraction, also increases. Now you can see three different lines here. The one in the middle is considered a control. So again, as you increase the amount of blood that's available in the atrium to be delivered to the ventricle for its next contraction, you have an increase in stroke volume and cardiac output. Then you see the lines above it and below it. These are the same Starling curves under the effect of either positively inotropic effects or negatively inotropic effects. So inotropic effects generally increase the contractility of the cardiac muscle fibers. In other words, a positive inotrope, such as cortisol, a stress hormone for example, increase the contractile tension of the cardiac muscle. In other words, the heart and the ventricle can contract harder and with greater strength. So under those conditions, the amount of stroke volume, the amount of blood that's pumped out per contraction for a given atrial pressure increases. Now, under negative inotropic effects, the contractile ability of the heart is decreased, which of course results in a lower stroke volume. And of course, there are conditions that would favor either of these two effects. So the Starling curve diagram here is just to show you that the ability of the heart to contract and push blood out is, number one, influenced by the amount of blood that remains in the atrium, but is influenced by external physiological. So if we, generally speaking, want to make it as easy as possible for the ventricle to deliver a high stroke volume, that means that we should increase our end diastolic volume. We should increase the amount of blood that's remaining in the atrium to be delivered to the ventricle for that next contraction. There's a couple of different ways to do that. So the first way is through filling pressure. This is the amount of pressure from the atrium. So pressure from the atrium can be a primary force responsible for actually filling the ventricle with blood for its next contraction. Now there's two different ways to do this, and they're referenced by their Latin terms here, visa tergo and visa front. Visa tergo is pressure that is initiated by the heart itself and then is carried through the rest of the circulatory system and returned to the heart via the venous system. So this is basically the amount of blood pressure that is quote-unquote left over after the blood has completed the circuit and is returning to the heart. As you might imagine, after completing the entire circuit, there isn't a huge amount of pressure remaining as the blood fills into that sinus venosus and then into the atrium. There's some pressure that drives it flowing into the atrium, but it's not much. So that's visa tergo, or from behind. That's what that visa tergo thing means, from behind filling pressure. Now visa front filling is filling from the front. In other words, it's cardiac suction that is created via contraction and release of the ventricle. So you can imagine that as the ventricle contracts and pushes blood out into the arteriosus, either the conus or the bulbous arteriosus, when it then opens and relaxes again, it can help to suction or draw blood from the atrium. And so this cardiac suction helps to increase the amount of filling and diastolic volume that is able to occur. So generally speaking, most fish that rely upon a pressure system heart, most fish that are highly active and more advanced, rely upon primarily visa front filling. In other words, that cardiac suction. More sluggish fish, more primitive fish, simply rely upon that residual pressure visa tergo filling. In other words, the kind of natural flow of blood back into the atrium after it's completed the circuit. A couple of things here to mention about filling time. Filling time is basically how long it takes for the atrium and the ventricle to fill up with blood after a contraction. Now, generally speaking, filling time doesn't usually influence cardiac output or end diastolic volume. Generally speaking, it's not a limiting factor. This is really only a limiting factor if the venous return is reduced. So if for some reason the amount of blood that's being pumped out by the heart isn't the same as the amount of blood that's being returned to the heart, it will take longer for the atrium and thus the ventricle to fill. Circumstances under which you might have a reduction in the amount of venous return would be if the fish sustains an injury and begins to hemorrhage or bleed out. That will reduce the filling time and will ultimately influence cardiac output. But again, generally speaking, under normal circumstances, filling time is not a limiting factor, in part because the heart rate of most fish is relatively slow. The typical heart rate for fish in beats per minute is usually between 20 and 60. The maximum beats per minute for most fish is less than 120 beats per minute. Now, this is, again, relatively slow. Keep in mind that the typical beats per minute for a resting human is between 60 and 100. And that's not that's at rest. It's not a human that's running, jogging, walking. It's just your beats per minute just resting there. So generally speaking, the beats per minute in fish are lower. The heart rate is lower. And so there's plenty of time, essentially, between contractions of the ventricle to allow that heart to be refilled. So again, filling time is not usually a problem. One other thing that can influence end diastolic volume is ventricular compliance. In other words, how stretchy is the ventricle? Stretchy ventricles or flexible ventricles tend to increase that end diastole volume, but they will have less capacity for visa front filling. In other words, a stretchy ventricle can be filled with more blood more quickly, but it's not going to be as strong in terms of sucking blood from the atrium using that visa front mechanism. So it's a little bit of a trade-off there. All three of these things, filling pressure, filling time, and ventricular compliance, will ultimately influence that end diastolic volume, the amount of blood that's available in the atrium to fill the ventricle for its next contraction, and that in and of itself, end diastolic volume, will influence cardiac output. Now, when you graph the cardiac function and the vascular function together, you can identify a balance between these two at a given physiological state. So what you're looking at here are two figures. So you have a cardiac function and a vascular function. So the cardiac function is basically the same thing as those Starling curves that you saw. So we have on the x-axis here right atrial pressure, or in the case of fish, just atrial pressure or end diastolic volume. So the amount of blood in the atrium after contraction of the ventricle. And then on the y-axis, you again have cardiac output. So there you can see the increasing dark heavy line, the cardiac output line. That's the same as the Starling curves that you've already seen. Now the opposite curve that starts high at the left-hand side of the figure and decreases is the venous return, or the vascular curve. This is the amount of blood that is returning to the heart in liters per minute, so it's still a volume per minute. The amount of blood that's returning to the heart as a function of that right atrial pressure. So when the right atrial pressure is low, there's a large volume of blood that can be returning to the heart. It's going down its pressure gradient from an area of high pressure in the venous system into an area of relatively low pressure in the atrium. However, as that right atrial pressure, as the atrial pressure increases, it can reverse that pressure gradient, and so you have a decline of the amount of blood that can flow into the atrium. Okay, so you have one curve that goes up from left to right, the cardiac output curve. You have another curve that goes down from left to right, that's the venous return. At any particular condition in the fish, the intercept will be the balance between those two. Ideally, you would have the same amount of blood going out from the heart as you would have returning into it. Okay, so at a particular physiological state, that intercept will represent a balance between cardiac output and venous return. Now the other thing that you see here is two dashed lines. These are also the cardiac output and venous return curves, but under greater TPR, or increased TPR. TPR here stands for total peripheral resistance. This is the amount of resistance that the blood encounters when it's circulating throughout the rest of the cardiovascular system. Okay, so as resistance increases, it compresses both of these curves. So for a given atrial pressure, you won't get as much blood out, and you also won't get as much blood back. In other words, it increases the total amount of time for blood to complete the circuit. Okay, so what does this mean for a fish? Well, let's think about some of the things that can increase total peripheral resistance. Decreasing temperature, for example, can increase the peripheral resistance. It does so by increasing the viscosity of the blood. As the blood becomes more viscous at colder temperatures, the total peripheral resistance increases, and so these two curves become suppressed. Okay, so you have less blood being able to be put out by the heart, so your cardiac output curve is suppressed, and your venous return curve is certainly suppressed as well. And so for a fish that's experiencing a reduction in temperature, that means their ability to complete the circuit of their cardiovascular system is going to be reduced. Or to achieve the same circulatory rate, they're going to have to expend more energy and either increase their heart rate or increase the strength of their contractions through perhaps a positive inotropic effect. Let's talk a little bit more about the intrinsic heart rate. The intrinsic heart rate of fish is set by the pacemaker cells in the sinus venosus and also in the atrium. So we talked about the sinus venosus being the primary location for the pacemaker cells. There are also pacemaker cells into the atrium. Okay, so the intrinsic rate, or the resting normal rate of the fish, is determined by those pacemaker cells, but as we said before, the actual rate at any moment in time can be adjusted up or adjusted down as appropriate. So tachycardia is an increase in heart rate. Bradycardia is a decrease in heart rate. So you can have tachycardic or bradycardic effects. Temperature is a good example. So increasing temperature results in an increasing intrinsic rate, or it influences the pacemaker cells to increase the heart rate. This should make sense because at warmer temperatures you have a higher metabolic rate, you have greater need for gas exchange, and you need to circulate the blood more rapidly. At colder temperatures, the opposite is true. Colder temperatures have a bradycardic effect. Adrenergic stimulation, this is stimulation by the sympathetic nervous system, is stimulatory, and it causes tachycardia. This is commonly what occurs when a fish is stressed or is exposed to a predator or some other type of threat. Cholinergic stimulation, or stimulation by the parasympathetic nervous system, has the opposite effect. So it decreases activity, it causes bradycardia, and brings that heart rate back down. Okay? So again, depending upon the circumstances that the fish is exposed to, its heart rate may need to change. Yes, it has a set intrinsic rate, but that can be influenced through these various tachycardic or bradycardic effects. Let's talk a little bit more about venous return and capacitance, or the total blood holding capability of the circulatory system. First, we need to talk about something called mean circulatory filling pressure. This is the pressure in the blood system, or in the cardiovascular system rather, at zero flow. Okay, so if we were able to stop the heart from beating and we allowed blood pressure to equilibrate throughout the entire circulatory system, that equilibrated average pressure would be the mean circulatory filling pressure. In other words, if we stop the heart of the fish and allowed the high pressure immediately downstream of the heart to equilibrate with the low pressure of the blood in the venous system and returning to the heart and the sinus venosus, if we allowed that to all average out after the heart was stopped, that would be the mean circulatory filling pressure. Okay, so in the blood system, in the bloodstream rather, the blood is under some pressure all the time, even if the heart isn't beating. So there's a certain amount of volume that's considered to be unstressed versus stressed. Okay, so the stressed blood volume is essentially the amount of blood that would flow out from the circulatory system if it were opened at that mean circulatory filling pressure. Okay, so if we were to take the fish, stop its heart, allow the blood pressure to equilibrate throughout the circulatory system, and then open one of the blood vessels, some blood would still flow out. Okay, not all of the blood would flow out, but a certain amount would flow out until blood pressure in the circulatory system equilibrated with air pressure actually outside the body of the fish. The amount that remains in the fish at equilibrated pressure is considered the unstressed or unpressurized blood volume. What does flow out to equilibrate that pressure is considered the stressed or the pressurized blood volume. Okay. The mean circulatory filling pressure will actually depend upon the compliance of the system and the total blood volume. So if you have stretchy venous systems, in other words, your capacitance vessels are very stretchy, that can reduce this mean circulatory filling pressure. In other words, the blood vessels will simply stretch to accommodate more blood and reduce that pressure. Keep in mind, again, that these venous vessels that are carrying deoxygenated blood, they're about 25 times more compliant or more stretchy than the arterial vessels. And this allows them to hold a greater volume of blood in the system and to have greater quote-unquote capacitance. Okay, so again, the venous system is much more compliant than the arterial system, and venous return and capacitance is going to be influenced by this mean circulatory filling pressure. Basically, how much blood is in the entire circulatory system and under. Okay, so in a resting trout, so a trout that's not swimming, not evading a predator, just sitting there, the unstressed volume is about 60% of the total blood volume. Okay, so if we were to stop the heart of the trout, allow blood pressure to equilibrate, open one of the blood vessels, 60% of the blood volume would stay in the fish. 40% of the blood volume would actually flow out. Okay, and that's at a mean circulatory filling pressure of 4.7 millimeters of mercury. Now, that's under normal resting conditions. If the fish were to be exposed to any number of different triggers, that would influence and affect the amount of stressed versus unstressed blood volume and basically the total blood pressure of the fish. Okay, so if, for example, there's an increase in something called atrial natriuretic peptide, this will decrease the mean circulatory filling pressure by decreasing vascular tone. In other words, it makes the vascular system, especially the venous vessels, much more stretchy. It increases their size, it dilates them, and as a result, it decreases cardiac output. Okay, it's decreasing cardiac output by making the entire system greater in volume, more stretchy. It's decreasing the amount of stressed volume. In other words, it would shift some of that stressed volume of blood into the unstressed category. Now, if, for example, there's an increase in blood volume, so a freshwater fish is stressed, it has an osmoregulatory failure, it starts taking on additional water, and the volume of blood in its system increases, that would increase the mean circulatory filling pressure. You have the same size of system, but more blood in it, therefore pressure would increase. So as a result, the fish oftentimes do release greater amounts of this atrial natriuretic peptide to compensate for that effect, because again, it has this reversing effect. It's going to decrease that pressure in response to increasing blood volume. Epinephrine is something that we commonly associate with stressful circumstances. it actually increases the mean circulatory filling pressure by increasing tone. So it constricts the blood vessels, especially the the venous system, which is going to shift that stressed to unstressed ratio of blood the opposite way. So under conditions where epinephrine is released, mean circulatory filling pressure increases, and so you have a greater volume of stressed blood as opposed to unstressed blood. So instead of it being 60-40, it might be closer to 50-50, or perhaps even 40-60. Why is this a good thing? Well, during a stress response, increasing this mean circulatory filling pressure will tend to increase cardiac output. Therefore, it's going to decrease the amount of time necessary to complete the circuit. It's going to increase gas exchange. It's going to make more oxygen, more nutrients, more resources available to a fish to support its fight or flight response. Okay. So again, there's a lot of different types of physiological or endocrine effects that can influence compliance or stretchiness of the vascular system. That can influence the mean circulatory filling pressure. These are just a handful of them, but again, I want you to understand that the vascular system is not a set, fixed entity. It is constantly changing its size, its stretchiness, and the pressure that it contains in order to maximize circulatory function at any given state. Now, generally speaking, there is a phylogenetic regression in blood volume. The more advanced the fish is, the smaller its blood volume. So for example, hagfishes, which would be considered a fairly primitive fish, typically have between 175 and 200 milliliters of blood per kilogram of fish. Teleosts, on the other hand, may only have between 30 and 40 milliliters of blood per kilogram of fish. So there's a dramatic reduction in the total volume. Why is this? Well, generally speaking, the more advanced fishes have more powerful hearts and can circulate blood more rapidly. So as a result of that, they can get away with using less blood in the system. They just circulate and recycle and reoxygenate that blood at the gills much more rapidly. So they don't need to have as much blood circulating at any one time. But there are other factors that are going to affect the total blood volume of a particular fish at a particular time. And again, keep in mind that the blood volume is going to influence that pressure, that mean circulatory filling pressure that we talked about, which of course is going to influence function of the circulatory system too. But taking this perspective, factors affecting blood volume, increasing red blood cell density and decreasing blood volume. Okay, so if you have greater numbers of red blood cells circulating, generally speaking, you don't have to have as much blood volume. This is because each unit of blood is more efficient at exchanging gases because it has more red blood cells and more hemoglobin for gas transport. If you have greater fluid exchange with the environment, in other words, you have really effective high surface area gills, generally speaking, you can get away with a reduced blood volume as well. If transcapillary exchange is more efficient, you also tend to have a decreasing blood volume. In other words, the more efficient you are as a fish in terms of acquiring oxygen, releasing CO2, exchanging materials at the gut and at the liver, and exchanging at the cells, delivering nutrients, picking up waste. The more efficient each of those mechanisms is, the less blood volume you need to accomplish those things. Okay? And again, remember that transcapillary exchange, or the rate of solute transfer, so you could think of this as the rate of oxygen delivery or the rate of CO2 pickup by the blood, is going to be a function of the capillary permeability, the surface area divided by the thickness of those capillaries, and the difference in the concentrations of the solutes. So if you have very permeable capillaries, they will be able to exchange solutes more readily. If you have a high surface area to vessel thickness ratio, that's also going to increase that rate of solute transfer. Again, more surface area allows greater ability for those materials to be exchanged with the cells. And of course, if you have a very large difference in the concentration of solutes in the cells or the tissues and in the bloodstream, that's going to facilitate that exchange. It's going to be a steeper concentration gradient, essentially, allowing for more rapid exchange at the capillaries. So again, the more efficient you are in terms of transcapillary exchange, excretion processes, fluid exchanges, oxygen uptake at the gill or CO2 release, all of these factors are going to drive blood volumes down. If, however, you are not very efficient at any of these things, blood volume will tend to be increased as a means of compensating for that reduced effectiveness of the circulatory system. We've already talked a little bit about regulation of the cardiovascular system via the pacemaker cells, but I want to talk a little bit more about this. So again, the autonomic nervous system is going to influence that intrinsic heart rate. So we talked about this already, the influence of sympathetic or adrenergic stimulation, which is tachycardic. It increases the heart rate and it supports fight or flight. Then in response to that, or countering that, we have the parasympathetic system, or cholinergic stimulation, which is bradycardic. It reduces the heart rate and is not supporting fight or flight, but is inducing rest and repose. There are other factors that can influence the heart rate as well. We talked about some of them already that act indirectly through influencing things like mean circulatory filling pressure. But there are other factors. There are, for example, baroreceptors or pressure sensors in the heart that are thought to help control the heart rate. So when pressure in the heart increases, the heart rate tends to decrease. When pressure is decreasing, however, the rate increases. So for example, if you have lower or reduced pressure, blood pressure in the heart itself, that's something of an indication that there's less blood being returned from the venous system. So that's probably not a good thing if there's less blood coming from the venous system. And so in response, the baroreceptors induce an increase in the heart rate to push more blood back in the hope that it will drive more blood to come back into the heart and complete the circuit. The opposite is very true. If you already have very high pressure in the heart and a lot of blood coming back to the heart after it's completed the cycle, it's probably not a good idea to try to push even more blood out into the circulatory system. So these baroreceptors result in a relative decrease in the heart rate. The gills themselves can also affect heart rate by influencing blood flow and back pressure on the heart. The gills are affected by the autonomic nervous system. So for example, the same cholinergic stimulation that we talked about in terms of decreasing the heart rate, it also decreases resistance at the gills. Okay, so it causes vasodilation at the gills. Adrenergic stimulation will have the opposite effect. It actually causes vasoconstriction during high activity at the gills. So these two factors are going to influence kind of the vascular compliance of the gills themselves and their ability to receive blood from the heart. Catecholamines, these are circulating stress hormones. Adrenaline, for example, is a very common catecholamine. These actually decrease resistance in the gill. So they increase the amount of blood that is allowed to flow through the gill lamellae, which again should make sense in the context of a stress response. If you're supporting fight or flight, you likely need greater gas exchange, and so by opening up and increasing the blood flow through the gills, it's going to facilitate that greater amount of gas exchange during a stress response. By opening up the gills and increasing their vascular dilation, that's going to decrease back pressure on the heart, again making it much easier for the heart to pump out even more blood and circulate through the system, which is also consistent with the needs of. So we talked about direct local regulation of the cardiovascular system by innervation, by the effects of the sympathetic or parasympathetic nervous system. But there are also remote effects, the endocrine system and hormones influencing cardiac function as well. So we'll just highlight a couple of these. For example, there's the renin-angiotensin system. This is considered a hypertensive action. So it's going to function to counter a reduced blood volume and reduced blood pressure. So if the fish has a reduction in blood volume being reduced in the same size of vessels is going to reduce blood pressure. And again, we talked about several times how maintaining the same level of blood pressure is important. So to counter that, that reduction in blood volume and reduction in blood pressure, renin, this hormone, is released from the kidney tissue. It causes the liver then to secrete angiotensin. Angiotensin results in short-term constriction of the systemic resistance vessels. So in other words, it's going to decrease the size of the vascular system, the capacitance of the vascular system, thereby increasing blood pressure in spite of this reduced blood volume. So that's the short-term effect. The long-term effect of angiotensin release is increased drinking behavior and uptake of water at the kidney. So this is actually going to increase that blood volume and build it back up to the higher blood volume that the fish originally had. Other remote effects include the natriuretic peptide. So we already talked about atrial natriuretic peptide. These function to counter the elevated blood volume and hypertension. So when blood volume increases, or for whatever reason blood pressure is too high, the cardiac muscle will secrete these natriuretic peptides. These decrease vascular tone, increase compliance or stretchiness, they decrease resistance throughout the entire vascular system, and they also increase the kidney filtration rate and they increase urination. So whereas the renin-angiotensin system serves to increase blood volume by encouraging the fish to drink water, to uptake water in the kidney, and it reduces the size or the capacitance of the vascular system, the natriuretic peptides have the opposite effect. They increase kidney filtration rates, allowing for greater release or excretion of water via urination. They decrease vascular tone, they increase the size or capacitance of the vascular system to counter an increase in blood pressure. A couple of others here, arginine vasotocin. This is a vasoconstrictive peptide that's released by the pituitary. Urotensin 2 is also another vasoconstrictor released by the urophysis, which is a part of the endocrine system of fishes. So there's a lot of different endocrine mechanisms as well as nervous system mechanisms to regulate some additional local factors that will influence the cardiovascular system. The production of metabolic byproducts, specifically production of CO2, or consumption of metabolic substrates, such as oxygen, will affect the cardiovascular system. Again, this makes sense since the whole function of the circulatory system is to manage levels of these two molecules. So if you have increasing CO2 or Krebs cycle intermediates, in other words, an indication of greater respiratory activity, you have decreasing pH and decreasing O2. All of these factors are vasodilatory. Okay, so if any of the tissues, muscle for example, exhibits an increase in CO2 availability, an increase in the concentration of Krebs cycle intermediates, it's experiencing a decrease in cellular pH, either intracellular or extracellular pH, and a decreasing availability of oxygen, that's an indication A little bit about the effects of exercise on the cardiovascular system. During burst swimming or swimming utilizing the white muscle, cardiac output is actually reduced in the short term, but then increased during recovery to clear lactic acid from the system. So initially, there really isn't a need for greater blood flow to the muscles initially because the white muscles don't really need that oxygen the same way that the red muscles do. But then, of course, the white muscle is going to generate a lot of lactic acid and CO2 that will need to be cleared before those muscles can be essentially reactivated. And so that's where you see this increase in cardiac output and functioning of the circulatory system during recovery from that burst swimming. With respect to sustained swimming or red muscle driven swimming, ventilation rates and cardiac output rates increase throughout the process. Again, this makes sense. So the red muscles require that supply of oxygenated blood, and so as a result, systemic resistance is decreased, so the vessels become dilated, flow to the muscles increases, whereas flow to the viscera may either stay the same or it may actually decrease. We talked about this as well. During swimming activity, digestive processes are less important than swimming to capture prey or to avoid a predator. So to maintain blood pressure throughout the system, let's talk a little bit about the effects of temperature. Decreasing temperature decreases metabolic rate and as a result decreases cardiac output. Decreasing temperature also increases viscosity of the blood, as we've discussed. To compensate for that, fishes that typically inhabit very cold environments where the temperatures throughout the year are exceptionally cold, they tend to have increasing heart sizes. This is to compensate for the difficulties in circulating more viscous blood. So the blood is more viscous, it's more difficult to circulate, so they have bigger, stronger hearts to try to compensate for that effect. Now, there is a trade-off at lower temperatures with respect to the number of red blood cells. So at those colder temperatures, the presence of red blood cells will lead to greater viscosity. In other words, the presence of high levels of red blood cells will make the blood much more difficult to circulate. However, there is usually less need for extremely efficient gas exchange. There's a reduced demand for red blood cells at very, very cold temperatures. And so the red blood cells give you more efficient oxygen exchange but greater viscosity, whereas on the other hand, if you have fewer red blood cells or no red blood cells, you don't have the viscosity problems, but you don't have the same efficiency with respect to gas transport. So I'm showing pictures here of two different types of ice fishes that inhabit polar environments. The so-called red-blooded ice fish do have red blood cells, and so they have to compensate for the viscosity of their blood by having relatively low vascular resistance. So their vascular system is quite stretchy, quite large. They have relatively large blood volumes, and then circulate and compensate in that way. Let's talk about the effects of oxygen availability. Acute hypoxia, in other words, short-term low levels of oxygen, will produce a reflex cholinergic bradycardia. Okay, so when oxygen becomes limiting, the heart rate in fish will commonly slow down. You can actually see the same thing in humans. If you're in a crowded room over a period of time, the air starts to get a little bit stale, oxygen availability begins to reduce, and as a result, people tend to get sluggish, their heart rates decrease, etc. Now, in teleos, blood pressure will increase as a result. In elasmobranchs, blood pressure will actually decrease. It's two different responses to this cholinergic bradycardia. In the case of teleos, their blood pressure increases. They have reduced cardiac suction. They have reduced cardiac suction. In other words, they have reduced visa front filling pressure. In the case of elasmobranchs, their pressure decreases because there is reduced visa ptergo pressure throughout the system. So two different responses to. I want to talk briefly about the secondary circulatory system. So this entire lecture we've been talking about the primary circulatory system, whose function, of course, is to deliver oxygen to the tissues that require it and to remove wastes that are produced by those cells and tissues. The purpose of the secondary circulatory system is a little bit different. So what is the secondary circulatory system? It's another system of vessels that delivers blood throughout the body, and it runs parallel with the primary circulatory system. The secondary circulatory system arises from the primary arteries, and eventually it will drain back to the primary veins. So it's essentially a branching off of the primary circulatory system. These secondary circulatory vessels are found in the gill, in the skin, in the peritoneal cavity, and in the oral mucosa, primarily in these surface or epithelial types of tissues. So epithelium on the outside of the fish, or the epithelial origin tissues lining the body cavity. Now, what do they do? What is their function? We know that it's not really gas exchange. Because we know that it's not gas exchange because there aren't red blood cells in the secondary circulatory system. There are actually guard cells, what are called microvillus guard cells, at the entryways to these vessels, and they act like little brushes, which essentially fence off the entryways. So red cells that are circulating through the secondary, red blood cells that are circulating throughout the primary circulatory system might try to go into the secondary circulatory system, but these microvillus guard cells brush them back and prevent them from entering. So because there are no red blood cells in the secondary circulatory system, we know that it's not, its purpose is not gas exchange. Now, because of the proximity of the secondary circulatory system to the skin, it's been suggested that there could be an osmoregulatory function here. It's also been suggested that the secondary circulatory system is responsible for nutrient exchange. In other words, distributing and exchanging things besides oxygen and carbon dioxide. So gas exchange of oxygen and carbon dioxide, we know that that's the primary purpose of the primary circulatory system. Since we don't have red blood cells in the secondary system, gas exchange isn't part of it, but the circulatory system does a lot more than just deliver oxygen and pick up CO2. So perhaps the secondary circulatory system is serving in some of that capacity. We also know that fish generally lack a lymphatic system. It's thought that the secondary circulation might be a precursor, an evolutionary precursor to the lymphatic system. The lymphatic system itself typically serves as a site of absorption of fats from the intestine. It assists in maintaining fluid balance. It's also what I like to call a garbage dump for the immune system. Antigenic, pathogenic materials that are identified by the immune system are often delivered to the lymphatic system, to the lymph nodes, for processing so that the immune system can more readily battle that same type of pathogen in the future. So that's kind of what the lymphatic system is typically supposed to be doing in organisms that have a proper lymphatic system. Fish, again, don't have that, but perhaps this secondary circulatory system is something akin to a lymphatic system. The secondary circulatory system may also serve to improve the efficiency of the primary system by removing most of the total volume from primary circulation. So it may be essentially a secondary circulatory tract that perhaps moves a little bit more slowly and is still functioning in a circulatory sense. It's still involved in nutrient exchange, but it doesn't necessarily need to circulate as rapidly as the portion of the circulatory system that's delivering O2 and picking up CO2. So if gas exchange is the primary, most important part of the circulatory system, perhaps this is something of a division of labor. So the primary system focuses on gas exchange, whereas the secondary system focuses more on the other exchange materials. And so by removing most of that volume into the secondary system, it increases the efficiency of the primary system. Again, it's not terribly clear what this secondary circulatory system does. It may function in all of these areas, but it is important to recognize that it exists in fish, even though we're not entirely sure what its function is. Okay, that is it for the cardiovascular system lecture. This is a kind of dense lecture. It provides a lot of information about a lot of different processes in the cardiovascular system, but you're probably wondering what is the most important elements that I need to take away from this. I expect you to be able to describe the